Hello, I'm Sanjana Nair and I will discuss with you my research titled An Integrative Approach to Decipher Viral Protein-Protein Interaction Networks and Their Structures. Viral infection is mediated through protein-protein interactions, and in our study we are predicting new protein-protein interactions through homology. The idea is that we have protein A and B that are known to interact, and if we have orthologs of A prime, and ortholog of B prime, A prime and B prime would be predicted to interact. So will A prime with B and A with B prime if these orthologous proteins share sufficient sequence identity with each other. Therefore, the hypothesis is that experimental information from one species can predict the putative PPIs in an ortholog. The way we do this is we take the target species, which could be virus itself or virus with the host, and we then mine these databases for interactions with the target species or the related species. If the interaction has been experimentally shown in the target species, the PPI is directly added to the network. However, if it has been seen in an ortholog, it is first mapped to the target species, and only if it fits some predefined criteria, it is added to the network. These interactions are then scored to assess the confidence levels. The PPIs are scored using these criteria with fluorescence microscopy and co-localization kind of experiments being scored lower than X-ray crystallography or direct interaction kind of experiments. Also, we take into account the number of publications, and higher number of publications reporting a PPI give a higher score to the PPI. Similarly, we also take into account whether the PPI is directly shown in the target species, that is experimental, or it is predicted through ortholog, and depending on the number of species in which it has been observed and the sequence identity between the ortholog and target species, we lower down the score. After this process, the ISMB intraviral PPI network looks like this, with 8 capsid proteins in blue, 25 tegument proteins in orange, 13 envelope proteins in yellow, and unannotated proteins in gray. These proteins are connected by lines that are the PPIs, with 433 PPIs, among which 271 are predicted, shown in red, 134 are experimentally determined in green, and 28 are experimentally determined as well as predicted through homologs in purple. The thickness of the line indicates the scores. Similarly, we have host viral PPI networks, for example HCMV, in which 144 HCMV proteins interact with 2400 human proteins and form close to 4000 PPIs. 3000 of these are experimental, shown in green, while 1000 are predicted, shown in red, and 19 are experimentally determined as well as predicted through homologs, shown in purple. We further found that tegument proteins form the majority of interactions with the host protein. Since this is so crowded and very difficult to analyze, we further clustered these networks. Shown on the left hand side is an intraviral cluster with the orange circles representing tegument proteins and the gray circles representing unannotated proteins. If we look at this cluster, we can speculate that the gray proteins would be involved in the process of tegumentation, since majority of the other proteins in the cluster have that function. Another thing we can do is we can take the viral proteins belonging to a particular function and try to assess which host proteins are involved in that function. So, for example, shown on the right hand side are capsid proteins, shown in blue, interacting with the host proteins, shown in gray, and we can specifically target the capsid assembly or capsid maturation process if we look at each of these interactions. But for that, we need to see which of these interactions are direct. And that can be done using AlphaFold. Shown on the left and right hand side are two complexes. On the left is tegument proteins UL19 and 35 shown in pink and blue, while on the right hand side you have UL16 with UL16 binding protein shown in purple and salmon respectively. How do we assess whether this is a direct interaction? We look at the PAE and the PLDT. PAE is the error and blue means that there is low error, and PLDT is the confidence AlphaFold has in that position being correct, and a high PLDT means it has high confidence. Therefore, a blue PAE and a high PLDT indicate that these interactions are correct and most likely direct interactions. Finally, I would like to conclude that our program predicts the interactome of all species, and clustering of the networks provides clues of interaction hubs, and structural modeling of the PPIs can be used to predict direct interactions. In the future, the program will be updated to predict domain-domain interactions.